All right. Good afternoon, everyone. My name is Winston Yan. I am a Termeer scholar and also now direct the Center for Genetic Surgery at the Broad Institute. It's a new initiative where I think the best summary of it is that just like Anna described, there's nodal biology, there's also nodal technologies. And what we can do is use those to repeatably and scalably treat many more patients, particularly those with monogenic diseases that we know the science can treat, but there may not be commercial opportunities. What I'm really here to do is introduce Hilary. And Hilary, she's had I would say, first an academic background that starts with Duke and with a postdoc at Harvard. Then she went into technology translation at Dana-Farber. That was her bridge into business development, where she has led business development at a number of storied gene editing companies, from Editas to Vor and Tome, and now is chief business officer at Profluent. I feel like Profluent, you can say that they are trying to one-shot develop and accelerate using AI new nucleases and new CRISPR editors to treat genetic disease with the right tool at the right time. So that's her professional background, but I think what's really special is here she is going to share a personal story. It's the first time that she's hearing it, so I think you're in for a treat, and I'm so grateful to be able to introduce her.
Funnily enough, I find myself a lot more nervous than if I was giving a science talk or a corporate presentation. It's funny when it hits home, so bear with me. But yeah, I'm going to share today, I think, about the intersection between professional and personal. And I'm going to start, with the slide of baby KJ, right? Exactly a year ago today was baby KJ's first dose of a custom personalized genetic medicine. And man, that took a village, right? You look at some of the reports in all the various journals, but also hearing from folks that were on the team, that was a lot of different folks that came together and collaborated in a sometimes can be rare way across for-profit corporations and not-for-profit labs. Of course, you have Kieran and Becca, the clinician scientists who spearheaded the actual treatment, but then you have manufacturing by Danaher, off-target done by the IGI Institute and Fyodor Ernov and Jennifer Doudna, the delivery vehicle coming from Acuitas, the alternative PAM coming from Ben Kleinstiver, just all these people who came together around this one baby who, if he hadn't received this treatment, would have almost certainly passed. So it's just sort of monumental. It's kind of, it's the dawn of maybe a new era. But that's just one baby. It's a proof point. It opens the door for regulators, for the media, for everybody looking at this story to say, oh, it can be done. This is possible. But ultimately, it took sort of a vat of midnight oil and many months and lots of dollars, often swallowed by the various collaborators at cost, to make this sort of breakthrough happen. And I'm going to come out and say that I believe we have a moral obligation to do this for all the other babies, right? And the question is, how do you scale? How do you turn it from a single proof point into something that is, more common? It's funny, you look at sort of the way that we do healthcare in America and contrast it with other countries. Somebody, sometime in our history, I don't know whom, I should probably read more of Heather Cox Richardson, but anyway, somebody at some point thought that it was worth providing and subsidizing healthcare for folks who are not well off and for folks who are old and no longer have an income, right? We have Medicare and Medicaid because people believe that all parts of society deserve equal access to healthcare. And I sometimes find myself wondering, why not a Medi-Rare, right? Why not an agency that exists to ensure that those that are most vulnerable from a rare disease perspective are not falling between the cracks? But then the question becomes, how do we do this, right? what needs to be changed to make baby KJ not a one-off breakthrough miracle, but sort of just the start of many babies. And so before I get into sort of the how and how we might disrupt the system, I'm going to talk a little bit about how I got to where I am and the personal meets professional part. So looking at my journey from scientist to patient advocate, on the surface, on sort of like above the water, it's an interesting track, right? I'm dating myself perhaps slightly, but when I got my undergrad degree at Michigan State University, the only way to get a genetics concentration was to do zoology. So I know more about big animals than I think I really ever wanted to know, right? But then I transitioned over to a PhD at Duke in cancer biology, and then came over here to Harvard and Dana-Farber for my postdoc on hormone-responsive cancers. And somewhere along that way, I kind of realized that what I got into science to do to help people, I wasn't sure necessarily whether I was going to be able to do that from the bench. And I became fascinated with this concept of how do basic research discoveries get turned into medicines? Because many of our research hospitals have absolutely fantastic basic science going on. The question becomes, how do you translate it? How do you pick the right partner to bring that, hopefully eventually to patients? And so I segued into some time in technology transfer at Dana-Farber Cancer Institute, and it was fascinating. I learned more about the science that was going on at Dana-Farber during my time in the tech transfer office than I did as a postdoc. And there's something kind of inherently wrong about that, right? When you're working in the lab, you can be so sort of head down that you know what you're doing. You maybe know what the labs next to you are doing, but when it goes further than that, you sort of get lost. So joined the tech transfer office, thought, now I'm going to learn all this business stuff. It's going to be great. And instead, I found myself on PubMed every single night being like, what are CAR T? I just got to sign the CAR T portfolio. How am I going to figure out how to license this? And then I moved over to my industry career, which has turned into a lineup of CRISPR companies. Ironically, when I first went to interview at Editas, I didn't even know what CRISPR was, right? This was very, very early on in the discovery. I had a few postdoc friends who were maybe using it as a screening tool, but I really had no concept of how that might be turned into an actual medicine. And so that as I progressed, through Editas, for Tome, and now Profluent, it was kind of a track through maybe the different generations, I guess you could say, of genetic medicine, right? CRISPR comes into the world, and it's amazing, and it also has certain limitations or certain mechanisms by which it acts. So then you have discoveries, many of which occurred right here at the Broad Institute, that find other methods of fine scale or larger scale insertion. And what was really interesting to me throughout this part is that, all of these companies are for-profit companies, right? They all need to somehow figure out not only how to help patients, but how to make money. And so, you'll do these clinical indication selection exercises where you're looking at all of these one-pagers of all of these awful diseases, right? Distilled down to a sheet of paper. Right? And trying to figure out whether the technology that you have in hand is going to be a good fit for fixing patients with those disorders. And it is about the patients, but it is also quite arm's length, right? The patient is other. It's not you. It's somebody else. And I spent most of the time that I was at Editas either pregnant or having babies, and that was it's interesting. You're sitting there and you're thinking, how does with all of the things that can go wrong in somebody's DNA, how are any of us normal, right? But it is very other. And so now I'll kind of flip to the other side of the coin, my personal life. So, whizzing through some of the early years, a lot of it was just moving for school and various parts of the career. But underlying all of this was the fact that since I was born, I experienced a lot of really weird, seemingly unrelated medical symptoms that no doctor could ever pinpoint. But it was all handleable or hackable or band-aidable, you know what I mean? It just that was my normal, right? And so, there we go. So then arriving at sort of the time that I moved to Boston and was spending time at Dana-Farber, I did marry a Brody. I want to put that out there. Matthew Eaton, he was an alumni from the Kellis Lab. And then we started trying to have babies, and we couldn't, right? And did all the tests, weren't any clear answers, got genetic screening, nothing popped out. And of course, this is genetic screening, a decade ago, so things have changed. And so we turned to science and the miracle of IVF. And in short order, had baby number 1 and baby number 2 while I was at Editas. And you know what it's like, any of you with kids and sort of like jobs, right? It's all just kind of a blur because you're juggling so many different things. And we kind of noticed in the beginning that like daughter number 2, Olivia, seemed to maybe not be kind of tracking the same way her sister did. But you go to the pediatrician and they say, all kids are different, right? Everybody has a different range. Everybody has don't compare between siblings. And then COVID hit, and everything shut down. And including many healthcare specialties. So a lot of the initial signs that we had that maybe something was going wrong just kind of came to a halt. But eventually we got to the point where Olivia was 28 months and she still wasn't walking. She still couldn't pull up to stand. We had been through the ringer. Folks were looking at, was it a dystrophy? Was it something neuro? What was going on? But everything just kept coming up nil. Until finally, one day we were in an orthopedic office, and the doctor turned and looked at me and said, "Hey, Mom, do you have any party tricks?" And I said, "I have lots of party tricks." "What are you talking about, though?" She's like, "Are you bendy? Can you move things in weird directions?" And I said, yes, absolutely. But I also walked when I was 9 months, right? So I don't see how that's really applicable to Olivia. And she said, but there are connective tissue disorders that we now know about, that we've been learning more about, and some of them seem to anticipate and get worse from generation to generation. She's like, so your experience as a child may not be, exactly translatable to your daughter's. And she was rotating Olivia's ankles, and she said, look, the kid's ankles can pronate 360 degrees. She's so flexible. And she was also a pretty fat baby, and so trying to develop the strength to match the extensibility explained why she wasn't walking. And so that was kind of the beginning of our diagnostic odyssey of, now it's we have a potential answer, go see genetics, right? And genetics said, well, unfortunately, we don't have a variant for this disease. So really the only way we can diagnose is through clinical symptoms. And because your daughter is not yet 3 and hasn't experienced a lot of life, can't really talk yet, the next best thing we can do is diagnose you. And if we diagnose you, then it's more easily extendable over to your daughter. And so that's exactly what happened. Again, it was still, coming out of the initial COVID shutdown. So waiting lists at most genetics clinics were anywhere from 1 to 3 years long, right? And when you have a 28-month-old that's not walking, you don't want to wait 3 years, let me tell you what, right? And so I begged, borrowed, and stole my way into a geneticist's office, and he took half a second look at me. No, not really, he took a lot longer look, but he said, oh my goodness, yeah, you have Ehlers-Danlos syndrome. And it was a name, it was a thing. And he said, it's great that you're doing this to get your daughters diagnosed. By the way, you're kind of fucked up and you should be seeing a lot of doctors. And I was like, oh, sure, yeah, but we gotta take care of the kid first, right? And so, that was, it was a really interesting sort of epiphany that sort of, I had something I didn't even realize I'd had my entire life. And then I inadvertently passed it down to my daughter. But over the course of the coming months, other stuff started to pop up that didn't really match anything in my health history. Unexpected bleeding, slow wound healing, and weird pain sensation, or actually lack thereof of pain sensation, constant illnesses. And again, you go into the pediatrician and they say, yeah, any kid that was inside during COVID, of course, now they're being exposed to all sorts of germs, and yes, they're gonna be sick all the time. And I said, I don't know about that though. The average course should be 7 to 10 days for a cold. And our youngest daughter is still sick 5 weeks later. And every week the school says, take her to the pediatrician, get a note so she can come back. And I'm like, it's the same cold though, why do I have to get another note, right? And so finally in 2024 and 2025, we managed to convince immunology to see us. And they did a first panel of blood tests that probably cost in the range of hundreds or less of dollars, so nothing super expensive. And it turned out that Olivia doesn't have B cell function and had no IgGA or IgAA. And that was kind of like the moment where like you get those test results back. Like I'm pretty sure I was like on a call giving the Profluent pitch, and I saw the patient portal ding on my phone. And like every doctor's nightmare, right? Because you're getting the results before your doctors even had the chance to look at them. And I'm not an MD, I'm a PhD, right? But I am dangerous enough between PubMed and ChatGPT to sort of like understand, right? It had been a few years since my last immunology course, but I immediately put everything into ChatGPT and was like, okay, we've got some answers here. Like, of course, if she, is not retaining or making or retaining any antibodies to anything she's been vaccinated against, of course, that explains why she got flu A 3 times the last year, right? But that kind of was a little bit sobering, because it's one thing to have a connective tissue disorder that is, affecting quality of life. It's another thing to look at primary immunodeficiencies. And so, that was the impetus that led to us being able to argue successfully with health insurance to get whole exome sequencing through Blue Cross Blue Shield and GeneDx. And when those results came in, surprisingly, they did not find a mutation in any of the genes that would typically be associated with some of these things. They instead found 2 more monogenic rare disease disorders. And you're just on the other side of the coin now, right? Like, no one-pager covering a disease prepares you as a parent to be in a doctor's office or to be at home on your computer, and all of a sudden you get an unexpected ding and you open it up and your whole life changes, right? Nothing prepares you for that whatsoever. And when you're a scientist, and my husband's also a scientist, you want answers, right? You want to know how do some of these de novo mutations correlate with phenotype? What is lifespan? What is severity? You want to know all of the data because that's the way that you were trained, right? And so that's actually what we've been doing. And I would note that like these latest diagnoses were only like 3-4 months ago. So it's still like, fresh. So, we're not at the end of our diagnostic journey. We're still sort of in it. So what's interesting to me here is that, when we tried explaining to the kids why they see doctors so much, it's hard talking with 2, 3, 4, 5-year-olds about what being sick or having a disorder and illness means, right? My older one, Fiona, kind of said, oh yeah, like when somebody has a broken leg, they have a cast, so they're sick, something's wrong, right? But you look at sort of like Christmas cards and stuff like that, the Facebook posts, and everything looks normal, right? Just 2 happy little blonde girls, right? But what's hiding underneath sort of that like veneer, right, are years where we have more than 150 doctor's appointments a year, right? Is Fiona, who's now 8, she has a connective tissue disorder, autism, and a rare congenital pain insensitivity, which I don't need to tell you is kind of a recipe for disaster on the playground because she can't feel temperature or cutting or bleeding, right? She has no fear because of the autism, and she craves sensation, right? So that's just, we're real friendly with a school nurse, let's put it that way. And then Olivia, who is now 6, was diagnosed with Oker-Chung neurodevelopmental syndrome, which Wendy Chung, the chief of pediatrics at Boston Children's Hospital, co-discovered as part of her efforts to sequence more kids and find more rare. The primary immunodeficiency, and as it turns out, 2 different connective tissue disorders, one of which I passed to her and one of which was completely de novo. And so here we are, right? This is Olivia now. This is our new normal. Once a month, we go in for a 6-hour infusion of other people's pooled antibodies in the hospital, which has been an absolute game changer. And for anybody who believes that science and medicine is not real, like that's proof positive, right? It's absolutely amazing, right? But this is kind of like what's lurking in the background, right? Some of these diseases, like there are, I think the latest count was somewhere between 360 and 400 kids diagnosed with Oker-Chung. So that's not very many data points, right? When you go looking for answers, which we did, we called up the rare disease nonprofit the day of the diagnosis and said, tell us everything. Where's the one pager on this? They don't have answers, right? They're doing their best to aggregate resources, but there just isn't enough out there to be able to say, this is what your child will experience. And so now then, juxtaposing that with my professional life, right? You can have the pity party, like, oh my goodness, how is this happening? How is it multiple diagnoses? But also, like, if two people were gonna have kids with rare genetic disorders, how about somebody who spent a decade at CRISPR companies and somebody who leads comp bio at a diagnostic startup? We were like, what can we do? Let's do something about this. And I just found it super ironic, right? That like I joined a CRISPR company not knowing how that was going to become a medicine. And now I have a daughter who could be a potential patient for a CRISPR-based therapy, right? And I remember the early days of CRISPR, right? Folks like Jennifer Doudna, David Liu, Katrine Bosley, the first CEO of Editas, they'd get up on stage and say, we have this new tool. We have something that is capable of fixing broken DNA. What would you rather do? Give your kid a pill that like treats maybe some of the symptoms but causes other symptoms every day of their life, or have one intervention that just restores their DNA to what it should have been in the first place? That, as long as it's safe, right? And CRISPR, when we look at it, the discovery at this point was a decade and a half ago-ish, right? So we've had a lot of progress for a new modality in a relatively short amount of time compared to what we've seen previously with other modalities. And yet the number of rare diseases that are out there, the number of genes that are linked to these conditions, the number of point mutations, right? It's so heterogeneous. Right? You've got diseases where there's a single point mutation that affects the majority of patients, but you also have rare diseases like my daughter's where pretty much most of the patients have every single one of them a different mutation. And not mutations that are even the same in terms of what they do to the DNA and the protein, right? Missense, splice variants, all these different ones, right? And it is really hard with many of the fine-scale editing modalities that have been developed to justify as a for-profit company, you can't make a single drug that cures all patients. So when you look at how much it costs to get something to an IND submission or a phase 1, how are you going to do that when you've got 100 kids with 100 different mutations and they aren't all addressable by a single drug, right? And so, one of the take-homes that I have is that like CRISPR is not sort of like one tool to cure them all, right? No single tool wins. We need all of the tools. Every single modality that exists within the editing space and beyond, all of them have a purpose. All of them have certain contexts that they are, compatible with potentially curing. One thing that has been also super interesting to me, particularly from the point of view of my current company, is that CRISPR, like many other medical breakthroughs that have helped lots of patients and made people tons of money, was not intentionally, quote unquote, discovered, right? Cas9 actually was found in a Danisco yogurt factory by yogurt manufacturers who were trying to figure out like how they could improve their fermentation processes. And then eventually, scientists figured out what it was that the enzyme was actually doing and how it could be co-opted to become a medicine, right? Insulin was discovered accidentally in a dog pancreas. And it completely changed the quality of life and the lifespan of kids with type 1 diabetes. Botox, legend has it that there was a band who consumed food from a dented tin can, and they all got super, super sick with botulism, and they experienced temporary paralysis. And some enterprising doctor or scientist realized, aha, we could use that for all of these other cosmetic and medical purposes. And when you think about sort of the world that we live in now, the fact that we rely on accidental discoveries and then try and figure out what use cases or applications we can apply them to, that's kind of horrific when you think about it. Not to sort of like discredit all of the amazing science that's going on, but like, we're at the point where it would be great to intentionally design things that are fit for purpose for whatever the use case is. And that's the wonderful thing, I think, about AI, right? There's been a lot of AI in the news. It's hard to tell sometimes what about it is hype and what about it is reality. It's a tool, though. It's a tool like any other tool, but it's a tool that has so much more capacity than one scientist or 10 scientists or 1,000 scientists, right? And when you sort of combine AI with the right people who understand the disease, who understand all of the elements of drug development, manufacturability, safety, like that is a sort of marriage made in heaven. When you look at sort of what has happened across the landscape of both academic and industry CRISPR and gene editing programs, one of the key themes is that we're somewhat limited by what nature gives us, right? So when something evolves in a germ to have a specific function, it has evolved under certain selective pressures to do a certain thing. And now we take it out of its native system and we're asking it to do something different, right? And so you can go about that in multiple ways, right? You can say, okay, this enzyme does certain things, I would like it to do more of those things or better of those things or slightly different things. And you can go looking in volcanoes and Arctic flows for more diversity of microbes that you might find different versions of these enzymes that have different criteria. But that's sort of a needle in a haystack, like whether you find something that's an exact match for your problem, not definite. You can do manual protein engineering, but the problem is that like when you look at a given protein, you're looking at a combinatorial space that's often more than the number of atoms in the world, right? 300 amino acids, 1,000 amino acids. Now times that, and it's just impossible, right? No amount of directed evolution, no amount of saturation mutagenesis is likely to get you where you want to go. And even if you manage to improve one quality of the protein, you often end up accidentally destroying another quality that you would have preferred to maintain. And with the help of AI, you can access some of that combinatorial space. You can try out things in silico that you may not have been able to do in the lab. And so the goal here is to not look for needles in a haystack, to not spend years, accidentally breaking proteins with a small number of mutations, but to design specific needles for each patient. And so how do you do that, right? How do you translate the success of AI models that have done so well with English language, with natural language, to protein language, right? So when you think about the success of ChatGPT, which did not exist when I was doing homework in college, but now is doing an amazing job at providing coherent essays and answers on every topic under the sun. Really, two of the main ingredients that led to that success were machine learning advances, like the transformer algorithm, and then also the availability of super, super large-scale data, right? The ability to go to the internet and just scrape billions of examples of English text for the models to be exposed to and practice with. And that's really the same thing that Profluent wanted to recapitulate on the side of protein languages, right? So you've all, or many of you may have heard of models like AlphaFold, right? Think of those as like readers of biology. You feed a protein sequence in, and it says, this is how I think the protein's going to fold, and therefore function, right? What we wanted to do was something different. We wanted to build a writer of biology, right? And we figured we had all of the elements for success that existed in the natural language processing world, right? Proteins are coded by letters of the alphabet, which resembles language. We have billions of examples of natural proteins that evolution has provided to us, right? Both dead ends and evolutionary successes, right? For the model to be exposed to. So the general idea then is you take a foundation model, you expose it to all of that natural diversity that exists, not just to the type of protein you're trying to make, but to all the proteins that you can get your hands on, right? And it plays sort of like trillions of these little games in a second, where it's sort of like fill in the blank, right? If you were like to say, a dog, what could come after that? If you say a dog kicked, what could come after that, right? So all of these little games, but the model's now playing it with amino acid sequences, right? It's learning sort of the grammar and syntax of protein language, right? Which domains do which things? What could you co-opt from a different family of proteins that might introduce a new function into the protein that you're trying to change? And once the model has been prompted with the exact criteria of what you want to build, so in a therapeutic use case, it would be, for example, I have a patient with this exact missense mutation. I need to fix it. I need to fix it in the liver. It's going to be delivered by an LNP. It needs to be this active, this specific. There are other A's nearby, so I need a really narrow base editing window to avoid bystander off targets. When you prompt the model with these things, it's not doing some sort of in silico version of mutagenesis. It's actually looking at the prompts, just like if you said, write me an essay about Shakespeare, and it's now writing millions of protein sequences, starting with amino acid 1, and then self-ranking them by which it thinks are most likely to achieve the outcome that you're looking for. And then you can test those in the lab. You can feed all of that lab data back into the model, and one of the glories of AI models is they don't really care whether the data is positive or negative, right? In science, you're often told that negative data is sort of like to be shoved under a table, particularly if you don't understand why you didn't get the answer you were looking for, right? But really, whether it's good or bad, the model learns something from it, and the next generation of sequences it produces are even better. So that's the general idea underlying how the process works of applying AI to protein language. And Profluent, since its inception in a research lab born out of Salesforce and OpenAI, has undergone sort of like a series of firsts in showing, can transformer models be applied to sequence generation in biology? Can LLMs learn underlying biophysics in an unsupervised manner? Can you take a small foundation model that's been trained on a relatively small number of protein sequences and then ask it to build a simple protein, zero shot? Can it do that successfully? And then now scaling into things that are more applicable to patients that we're talking about today, can you, for example, ask your model to create a Cas9 lookalike? And does it function? And the answer was yes, and we open sourced it. It's available free for anybody to use. For research commercial uses, we don't care, because it was just a proof of concept of what can these models do when you prompt them with various tasks. The next thing to come up was, does it matter how big the model is, and does it matter how much data you feed the model? And the answer is yes, it matters very much. Bigger is better in this case. The more data, the better. The more examples of proteins and the more metadata that you feed the model, the better it gets at being able to not just interpolate, but also starting to extrapolate and go beyond what it's been provided with, right? And then finally, and relevant particularly for gene editing, right? It's not just the protein. No protein exists in a vacuum by itself, right? There's all sorts of interactions going on. And so can you get to the point where you could accurately predict how all of those interactions are going on so that you take that into account as you're engineering your proteins? So then how does this fit into the concept of democratizing gene editing with AI? Why does this matter to patients, right? How do we disrupt the status quo? I don't know if I've covered all of the bottlenecks or obstacles that exist in the world of getting sort of gene editing treatments to rare disease patients, but boy, there are a lot of them, right? The regulatory path, at least before many of the folks in this room got involved with plausible mechanism pathway, is too lengthy for many rare disease patients, right? The patient populations are so small that many for-profit companies look at it and say, I can't justify this to my investors. We're not going to get any return on investment, right? Trial timelines are too long, right? In many of these diseases, we're talking about, kids that if you don't intervene, in the first months, first year of life, then it's too late. There's too much deterioration, right? Manufacturing cogs are super high, right? It's justifiably hard to make sure that you are making safe material to put into human babies, and some of the things that have applied in large populations of milder diseases where the severity is not quite there, do all of those need to apply to some of the rare disease trials that we do? And then finally, a lot of it comes down to economic feasibility, right? In this country, in other countries, how things are reimbursed is huge. And frankly, when you look at sort of the financial royalty and milestone stack of having to combine together all the different complex elements of some of these therapies, you end up with therapies that no big pharma can look at and justify selling because you're not going to make a profit. So I am not suggesting that AI is the panacea that fixes all of these things. It's not, right? But there are certain places very early on in drug development where it absolutely can make a difference, right? First is the trial timeline. When a baby like baby KJ is born with a specific mutation, if you have a model that is capable of spitting out a bespoke editor that is safe and efficient and tuned precisely to that baby's mutation in a matter of hours, right? That's a lot different than some of the timelines that we would work with in platforms at previous companies, right? And then economic feasibility is sort of another area around sort of like if you can find ways to create novel molecules that have freedom to operate and then utilize those in the rare disease space, right? You can start finding a path to sustainability on the economic side. I also think that there could be a lot more collaboration between players across the ecosystem, where often sort of like each person's fighting for their part of the pie. But when you get folks together like they came together for baby KJ, you stop thinking about profit margins. You start thinking about ways to creatively make things work. That's where the true magic is. So I'll end by saying rare is not actually rare, right? 300 to 400 million people in the world. And that's just based on the diagnostic criteria we have today. If every single baby was being whole exome or whole genome sequenced, I bet this room would light up. No offense, right? And so it's not just rare, it's not just me, it's probably you too, right? And one miracle was just the beginning, right? Now we need to start focusing on building the system that delivers the next million and does it in a way that's sort of agnostic to socioeconomic, geographic location, how many patients are in the disease, right? Everybody deserves access to a healthy life. So thank you again to the organizers for having me, and yes.
Thank you so much. I think that was just an incredible talk that highlights the key point of when people ask like, oh wait, 1 in 10 people have ultra rare disease or rare disease. That's a shocking number. I've known you for almost 10 years now, right? Like I would not have known this sitting across the deal table from you. And I think that's just a great lesson for us, that it's what lies beneath. There's this incredible impact of rare disease on all of us, and it may not always be obvious, especially when you have someone as eloquent as you. So thank you for sharing that story. I think that's incredibly, I would love to give Hilary another chance. That was the first time you shared it. So we have time for 1 or 2 questions. Please, can you go up to the other microphone over there?
Hilary, thank you for sharing your story, and I'm saddened to hear that as a mother, as a parent, that you have to manage that with your child. And I'm not much of a scientist. I have background having run this former hospital's research department, so I'm more of an administrative person. But what I've been noticing is a lot of the symptoms that would be considered of rare disease, and I'm not saying like your kid doesn't have rare disease, are coming up more in children who wouldn't have it because of like environmental issues, especially like, for example, chlormecrat, CHLORMEQUAT. which is causing like a lot of the symptoms that you see that gets displayed in a lot of those rare diseases, like which is like in a pesticides that's put like in Cheerio that people give their babies and that's making them sick, and food coloring. Like, have you done any of those type of like removal of those like harmful food from your, like, I wonder like what has diet and those type of like environmental changes have played in like the cure besides just like giving people medication? Have you seen any effect of that? I don't know if you've done that.
Yeah, so that's actually an amazing question. I think what we're starting to understand, that it goes back to sort of the nature versus nurture, right? Everybody has unique genetics. Everybody's also exposed to the environment, right? And I think what we're starting to learn, and it gets back maybe to some of the nodal research that is in your paper, is this idea of, you can be exposed to things in this world, and they could be environmental triggers like pesticides, or they could be viral triggers, right? COVID or Epstein-Barr or Lyme disease. And if you have the right genetic background, you might be even more susceptible to your body entering some sort of neuroinflammatory or other inflammatory state that then makes an underlying diagnosis that would have been fairly mild much more pronounced. I think the more that we can find information about some of this stuff, the more that we'll find treatments that aren't necessarily aimed at a single gene or a single mutation, but we'll find things that will sort of, buffer or restore somebody's genetics to, somebody with a different background who wouldn't have such an overt reaction to some of these triggers.
Hi, Hilary. Good to see you again. I'm Ken Leclerc. I met Hilary at Editas when we sat across the hall from each other. And I had just had my daughter, who's otherwise normal, develop all sorts of hypermobility, joint displacements, and we talked about that. And then, so she was diagnosed at Children's with Ehlers-Danlos. And small world, in the office next to me, our chief medical officer heard us talking or something and said, "Oh, yeah, I have it too." So as a scientist, you can imagine what's going on, what just happened to my beautiful girl. So I founded a foundation, and I got genome sequencing, which at that time didn't really, this is back in 2012, didn't really disclose much. But that also pushed me to look at the National Ehlers-Danlos Society. Claire Francomano down in Baltimore is wonderful, clinician. And with some connections here, I was able to connect Joel Hirschhorn, a brilliant geneticist, to the National Ehlers-Danlos Foundation. And they've just completed their HEDGE study. I don't know if you heard of that.
I was at the conference last year, yeah.
Lovely. So hopefully, they'll be able to find out what affects people in families with Ehlers-Danlos. I did our genome redone at sequencing.com last December, and with AI prediction of the variants, oh my God, Ehlers-Danlos popped up in her profile. So and the molecular cause is now understood. So just good luck to you, and I'm going to catch up with you at the break, I hope.
Thanks, Ken. And it just goes, the final note here is, it takes a village, right? What you're hearing here is basic research, clinicians who have a passion for connecting that with discoveries, admins and other support folks who are helping the entire machine continue to work. You don't know who it is that's going to touch you and that's going to make the discovery or connect the dots that lead to something that actually impacts your life. So thank you. I will clap for this room and say thank all of you for everything that you do. It's